We are talking about extracellular matrix, and we have been talking about the different functions extracellular matrix can perform beyond being just a structural element. In this module, let's talk about how ECM, extracellular matrix, can influence cell shape, survival, and proliferation. So let's look at that. If cells are grown on organized fibronectin fibril matrix, they flatten out and assemble intracellular stress fibers that align with the extracellular fibronectin fibrils. In one of the previous modules, I showed you a very nice micrograph in which there were two molecules, fibronectin and actin. They were stained or they were colored by different dyes, and we saw how they lined up. So the reason why they line up is because one influences the other, and this interaction is reciprocal. So here, for example, you can see in this photograph that here, for example, are the molecules of ECM colored in red, and they are lining up with the cytoskeletal elements which are right underneath the cell. Of course, you cannot see in this case because cell is sitting on top of these fibrils made up of extracellular matrix. Basically, one element can influence the orientation of the other element, meaning ECM can influence the cytoskeleton, and in turn cytoskeleton can influence the ECM orientation of ECM fibers. For example, here we have a cell. We place this cell number 1 on ECM, which was organized into fibrils. So these fibrils are sitting on a culture dish on which you have placed these cells. This cell number 1 only. These other cells 2, 3, 4 and 5, they are not sitting on any ECM. So you can see their cytoskeletal array is disorganized. For example, in 2, 3, 4 and 5, their cytoskeletal array is disorganized as compared to the cell number 1, which is sitting on the organized matrix molecules. So once this cell sits on it, it is going to produce new molecules, secrete more matrix molecules, and those matrix molecules are secreted. They will also line up in accordance in the same orientation as that of the cytoskeleton of the cell. So when this cell produces new matrix molecules, it is going to line them up in the same orientation as its cytoskeletal elements are lined up. So when this ECM, the cell is producing, cell number 1 is producing ECM in the dish, when these other cells, for example, cell number 2 and cell number 3, when they come in contact with the ECM, which is being deposited by cell number 1, they will interact with this ECM, and there, as you will see in the next slide, their cytoskeletal elements will also now line up in the same orientation, in the same way our ECM molecules are. Here, now you see that cell number 2 and cell number 3, when they came in contact with the matrix molecules which were produced by cell number 1, their skeletal elements, their cytoskeletal elements have also become organized, and their orientation is similar to that of the ECM molecules. So cell number 2, cell number 3. Now these cells, cell number 2 and cell number 3, they are also going to produce ECM, and when they produce ECM, it is going to be in the same orientation as was their orientation of their cytoskeletal elements. Let's move on to the next slide. Here, for example, so this cell made ECM, which lined up the cytoskeletal elements of cell number 2, cell number 3. These cells 2 and 3, when they produced ECM, it was of a similar orientation of their cytoskeleton, and when this ECM was produced, it caused the skeletal elements of cell number 4 and 5 also to become organized and also become lined up with the ECM they have secreted. So a cell can influence its neighboring cells. It can influence the structure or the shape of the cell by secreting organized ECM molecules. Okay. Most cells need to attach to ECM to grow and proliferate and also to survive. This is referred to as anchorage dependence, and it is mediated mainly by integrins which generate intracellular signals telling these cells to live. If you remove these cells, if you take the ECM away from these cells will not be able to survive. An additional feature of this interaction is the physical spreading of the cell on ECM. It has a strong influence on the intracellular events that allows cells to spread over a large surface area. They survive better and proliferate faster than the cells that are not so spread out, even if the amount of interaction between the cell adhesion molecules and ECM is the same. So let's look at this experiment. Here you have X amount of ECM, fibronectin say. The X amount of fibronectin is placed on a surface of a petri dish or a tissue culture dish. And in one case, we have it in a form of single unit. It is a single drop of fibronectin. And we take the similar amount of fibronectin. And now rather than making one big area of fibronectin, we make several smaller areas. Now, please remember the amount of fibronectin is same. It is just distributed differently. And we put a cell on this fibronectin, and we also put a cell in this area. The result, the cell which was put on this area, the amount again of fibronectin is the same. This cell died via apoptosis. It could not survive. Whereas the cell which was put on this area, this cell survived. So it is not the amount of interaction. The ECM also has to allow cell to spread itself. So there's more to just the quantitative relationship of ECM and adhesion. Additionally, ECM should also give cells an opportunity to spread themselves. If they are not allowed to spread, cells can go through apoptosis or programmed cell death.